PyMOL is a molecular modeling software used for making publication quality renders of biochemical structures. On screen are just a few examples of what's possible by utilizing PyMOL. This video is the first of our PyMOL tutorial series on YouTube. In this series, you will learn how to become proficient in the PyMOL interface and create high-fidelity renders such as the ones previously shown. This tutorial is also available as a Google Doc in the description down below. In this video, we will cover first the user interface, and then finish up by creating a simple yet high-quality visualization. The installation of PyMOL will not be covered in this video, as we have previously made a video on how to install lots of software including PyMOL, which will be available in the description down below. Let's begin. One of the first things you might notice when you open a new PyMOL window are the tabs at the top of the screen. These tabs contain the settings for PyMOL and make up what is called the UI, or user interface. I want to quickly break down what each of these tabs do, as they contain some useful settings for further use. The first tab is called File. Similar to most softwares, this is used for opening, saving, and exporting files. When you click on it, these options appear. When performing visualization, it is sometimes useful to have multiple windows open at one time. By clicking New PyMOL Window and Default, you will have two different windows open, and you can multitask, which is incredibly useful, especially if you're comparing two structures at one time. The next option is Open and Open Recent. As the name would imply, this simply opens files or sessions for PyMOL. The next option is Get PDB. When performing work for proteins, you may often memorize the PDB code associated with that protein. This is very useful because you can simply type in the code. So in this case, I'm using 4PRG, which is a PPAR gamma, and I can type in the chain. Now the chain are the different subcomponents of the protein, so in this case we're just going to go with A and hit download. And as you can see, the protein is downloaded right here. The next option is Save Session As and Save Session. As the name would imply, these allow you to save the current PyMOL session. Now I keep bringing up the word session, and you may wonder what that is. A PyMOL session is simply the current structures and settings loaded into your window, and you can save that as a file to go back and continue your work later. The next option is Export Molecule. This is useful if you've tinkered with a molecule and you want to save it as its own file to manipulate in another program or even in another PyMOL window. So by clicking Export Molecule, I can go down to my selection and I can select 4PRGA, which happens to be this molecule right here. So let's say I removed the ligand and I wanted just the protein, the receptor. I could type in 4PRGA, hit save, and save this as a file. The next option is Export Image As. This isn't used a ton because we'll use draw and ray mostly, and I will get into the differences here in a little bit and show you how we use draw or ray, but this will simply save the current viewport you have of the protein. Draw and ray gives us more options, and once again we will use that further in the tutorial. Export Movie As is a lot similar to Image As. The only difference is you're exporting a movie, and there really isn't many options in terms of using an alternative setting here at the right. The last thing we're going to go over is Reinitialize. We're going to skip over these tabs right here, as some of them contain some options that are a little bit more advanced than we want to go into currently. The next tab we're going to investigate is Display. Now Display changes the settings and options for the viewport in PyMOL. The viewport is what I'm referring to right down here. It is also sometimes called the preview window, and I may use those two terms interchangeably. So if we click Display, the first setting we have is Sequence. This is by far one of the most useful options, as you can see it displays all the residues present in your current structures. So for a protein, this is going to be amino acids, such as this scenario, and for DNA, it's going to be nucleic acids. So as you can see here, it's displayed by one-letter codes, and some people find that a little challenging to read, and so one of the other useful setting in Display is Sequence Mode. Now you can choose this and then change it to names, and this will allow you to look at the different three-letter codes for the different amino acids, which a lot of people find a lot more useful and a lot more legible. We're going to skip down to a few options here and go down to Background. This setting allows you to change the color of the background. Now for publications, it is not recommended to use black backgrounds. The reason is the high contrast view between some of the colors you use and black isn't quite as aesthetic as what's necessary for a publication. In addition, if the publication is printed out in red on paper, black can consume a lot more ink and make it harder to view in general. So by clicking background and then changing that to white, you can now see the background is white instead of black, which is a lot more useful to us. The next setting we're going to look at is quality. Now what this does is change the quality of the preview window in PyMOL. So if I want to change this to maximum performance, you can see on your screen, and actually you can adjust the viewport by using this bar right here. The quality decreased just a little bit. Now if I go back to display, quality, and do maximum quality, you can see it blurred it a little bit, made it a little bit more defined and easier to see, and maybe some anti-aliasing going on. Now for your system, you might want to change it to maximum performance if you're having trouble running PyMOL, but if you want to get a better preview of what the render will look like when you finish it, I recommend staying on maximum quality or reasonable quality, so it gives you those options for performance. The next one we're going to look at is color space. So color space changes the colors that PyMOL uses to represent things. So right now by default you can see that we have this green, it's kind of a neon green. So if I hit display, color space, and change it to CMYK, you can see it tones it down a little bit and changes the hues of the green. Now this isn't something you necessarily have to use every time, even though this says that it's used for publications, but it does give you a little bit more options if you want to try out some different colors and some different art styles, as making a visualization does have a little bit of an artistic value to it. The next option in the display tab that we're going to look at is grid. So if I hit display, grid, you can see I have a couple options here. The first one is by object. If I choose that, you can see nothing happens. Similarly, if I choose display, grid, and state, nothing happens still. Now if you look at my project table, this actually makes sense. So the only object I have loaded in is 4PRG. In addition to that, it only has a single state, meaning it only has one energy position located in its PDB file. So what I'm going to do is I'm going to get a PDB in here just so we can compare the two. So I'm going to hit File, get PDB, and I'm going to use the same 4-letter code as before, but this time I'm going to compare chain B and hit Download. So as you can now see, I have two chains loaded in here. So if I hit Display, Grid, and by object, at first it just looks like the two are separated, but if I actually play around with it, you can see I'm rotating the two different chains individually of each other. In addition, the spacing allows for better comparison. This is incredibly useful if you're comparing different chains of an object or comparing two different mutations of one thing. For now, I'm going to turn off the grid option so we can move on to the next step. Next is the orthoscopic view. If I hit display orthoscopic view, you may notice the preview window will warp slightly. And if I hit display orthoscopic view again to turn it off, you'll notice it warps back. Now what this does is slightly distort the screen in order to allow further understanding of depth perception. When you have complex structures shown in the preview window, it can get a little challenging to understand the depth of objects. Having orthoscopic view set to on will aid in that. Now I would not recommend you leave it on if you are making a render, but while you are viewing something, it is beneficial to keep on. The next thing we're going to look at is show valences. So if I zoom in on this ligand down here on chain B, you can see little lines in this ring. In addition, you can see little lines near this oxygen. These are the valences of the ligand. Now, they are representative of the chemical structure, but it is not recommended you keep these on while you are making a render. Instead, hit Display, Show Valences, and turn them off for a smoother look. The final option we're going to look at in display is Depth Q. If I hit Display, Depth Q, you can see some of the areas of the protein cleared up a little bit and have less fog. What Depth Q does is add a little bit of fog on screen to aid in further depth perception. Now, whether or not you keep this on is up to personal preference. However, if you are making multiple renders, I recommend that you pick whether or not you want depth Q on at the beginning so that you do not have some renders with fog and some without. In addition, if you can scroll your wheel, you will notice that the fog will go away on its own. If I scroll back down, you can see I add fog. And if I scroll far enough down, you can see I start to take away and peel at some of the protein. This can be useful if you wanted to say peel back some of the structure and look just at the ligand. The next tab we'll look at is the Setting tab. Now, this houses all of the options for adjusting representations of structures on screen. When I say representation, I mean that this protein, for instance, is being represented by cartoon ribbons, tubes, and beta sheets. In addition, the ligand is being represented by sticks. So if I wanted to adjust that, I could use the Edit All setting. So by hitting Setting, Edit All, I could change the cartoon radius, or I could change the line radius. There's a lot of different options I have within here. Now, I don't recommend you spend a lot of time here as a beginner, but if you're interested, this will be covered further in advanced tutorials. The next thing we're going to look at in the Setting tab is Colors. Colors allows you to adjust the default value for different colors within PyMOL. For instance, if I wanted to change black and make it just a little lighter, more of a charcoal, I could do that by adjusting all of the red, green, and blue values. Arguably more useful, if I wanted to change the default color for an atom type, I could do that. For instance, if I have a phospholipid bilayer and I want to change the color of the phosphorus atoms, I can click phosphorus and I can adjust it. So by dragging this up and this down, so green up and red down, and then blue up just a little bit more, I can make it more of a tan color and then hit apply. And then from there, the default color for phosphorus is going to be this. The next thing I'm going to look at in settings are a couple different options at one time. If you look at label through transparency, you might notice some similar options within these to the Edit All option. The reason is because this is essentially a condensed version of the Edit All. Now, I can still adjust certain things such as stick radius, but I can't set my own value. Instead, I have to choose from a preset. This is very useful in a pinch or if you're a beginner. However, if you want to do some more advanced tweaking, click the Edit All tab. The next thing I want to go into is Auto Zoom New Objects. So by default, when you load in a new object into PyMOL, it will zoom to show everything at one time. That can be a little frustrating if you have a specific view that you want to replicate for multiple renders. So for instance, if I like this camera angle and I load in a new object, it's going to adjust that. So by hitting Setting and Auto Zoom New Objects, we can turn that off. Similarly, when you load in new objects, you have the option to auto remove hydrogens or not. So you'll see this further in the tutorial, but one of the first steps in making a good visualization is to remove the hydrogens. So I recommend you check this, as it will automatically strip them and you'll save some time later. Now the Scene tab. The Scene tab houses the options that let you store camera angles and perspectives of the representations as scenes to be retrieved later. For instance, if I wanted a side view on this chain B that's yellow, I can simply hit Scene, Store, F1. You'll now notice an F1 in the bottom left corner of the screen. And if I wanted to store a view of the green chain A, I can hit Scene, Store, F2. Now I can cycle through by hitting Previous or Next under the Scene tab. I can also cycle through by hitting the Page Up or Page Down buttons on your keyboard. Or I can hit the buttons on the left-hand side of the screen. By hitting Scene, Append, it will add a new scene as a default number on the bottom left side of the screen. By hitting Scene, Insert Before, it will insert a scene before the current scene we are on. By hitting Insert After, it will insert a scene after the current scene we are on. By hitting Scene Update, it will change the current scene we have clicked. So as you can see now, I have scene 001 set, and it will change it to what is currently set. So hitting Scene Update will change 001. By hitting Scene, Delete, we can delete the current scene we have. By hitting Scene, Recall, we can simply retrieve the F1 through F12 scenes. And by hitting Scene, Clear, we can clear one of the scenes that are set to the F1 through F12 buttons. The next tab is the Mouse tab. This tab houses all of the options that changes how your mouse interacts with the screen. The first setting on here is Selection Mode. By default, it is set to select residues. For instance, if I click on this protein, I select all of the residue atoms that are in the location I clicked, not just the single atom I'm clicking. This may not be preferable. If I want to change this, I can hit Mouse, Selection Mode, and Atoms. Now, I will select only the atom that I click. Additionally, I can change the selection mode to Chains, which will allow me to select an entire chain at a time, such as chain A and chain B. For now, I'm going to set it back to the default residue. The final tab we will go over is the Wizard tab. This tab contains a set of tools that helps us complete very specific tasks within PyMOL. For instance, the first option in here is Appearance. There are a couple options within here that allow us to toggle, color, show, or hide things in the representation. Right now I have it set to Toggle, Cartoons, by Residue. So if I click this residue, it's going to toggle the cartoon off, which is pretty useful, especially if you're trying to hide certain aspects of it. Similarly, I could color it. So we're going to color magenta by residue. So now I can color certain aspects of the protein. When I'm done, I hit Done. The next tool within Wizard is Measurement. This is one of the most useful tools within this. What this allows us to do is get a measurement in angstroms from one atom to another. So right now, let's say I want to measure the distance between this oxygen right here and the residue here. I can click on that, and boom. It's measuring 5.4 angstroms between this oxygen and this backbone. When I'm done, I hit Done. Very useful. The next tool within here is the Mutagenesis tool. Now I won't go into this in detail, but this will allow you to change the certain residues / settings of the residues within the atom. I'll cover this further in a special tutorial, but for now, just know that it's present. When you're done, once again, hit Done. The next tool within the Wizard tab we'll cover is Label. This allows us to very quickly label certain aspects of the protein. By clicking the wizard, you can see it opens up at the bottom right. We have a couple different options for the mode, but for now we'll leave it to label the residue number and the residue name. By clicking different residues on the protein, you can see the name will appear and the number associated with it. When you're done, once again, hit Done. The final thing I want to show you in Wizard is the Demo button. This gives you a series of demonstrations to play around with some of the mechanics available in PyMOL. For instance, if I hit Demo Representations, it displays all of the different representations available in PyMOL, from cartoon to ribbon to dots to mesh, etc., etc. You can cycle through all these demonstrations on the right. One of my favorite is sculpting. This allows you to control click on this molecule to play around with it in a 3D space. It's very fun. I highly recommend that a new user plays around with lots of these options to see what's available and see the scope of PyMOL. Now we will apply some of what we learned in the user interface section to make a visualization of a structure called 1-EMA, a green fluorescence protein. Biomolecules like this exist in the real world as 3D entities. Scientists can capture a 3D model of these entities and export them to a digital format. In this case, we're using a protein databank file, otherwise referred to as a PDB. There are a couple different formats that PyMOL can take, but PDB is by far one of the most common. Similar to before, we will use the File Get PDB option to retrieve the PDB directly. Alternatively, if you were browsing for a PDB, you could open a web browser and go to the rcsb.org website, which I will link below, and manually download a structure to open using this option. However, since we know what we want, we can just use getPDB. Type in 1EMA, and this structure only has one chain, so we don't need to specify that. Now we can hit download. You should now see the protein at the bottom. The first thing I'm going to do is drag this window up just to make it a little larger, and then I'm going to show you how to use the mouse controls. Now these controls will vary depending on your system. If you're using a trackpad, it's a little bit more challenging, and I recommend you use a mouse. For the purposes of this tutorial, I will show you how to use a mouse. So left clicking in the center area of the protein will allow you to rotate. If you left click on the outer edges, you will rotate only around the Z-axis, which is the axis that's pointing straight at the camera, straight at you. If you right click, you can go up and down to zoom in or out. If you have a mouse with a middle mouse button, you can click on the middle mouse button and drag to move around in the XY plane. And if you scroll with a mouse that has a scroll wheel, you can peel back more or less of the protein by scrolling up or down. Now, if you are on a device that does not have a scroll wheel or a middle mouse button, what you can do is hit Control or Command and then click, and it will allow you to drag around the XY axis. One of the first things to do when starting a new PyMOL session is to change some initial settings. The first thing to do is to set the background to white. This can be done by hitting Display, Background, White. As previously stated, this enhances the visibility and makes the image better for publication. The second thing to do is set the valence to 0. As you can see here on the ligand, there are little lines that are a byproduct of the valence being set to on. If we set the valence to off by hitting Display, Show Valence, it will remove these. This just enhances the view and makes it look nicer. The next thing we're going to do is set the Cartoon Sidechain Helper to on. The Cartoon Sidechain Helper is a tool that allows for better visibility of residues. For instance, if I was to select a residue right here and then show it, you can see the backbone and it points out. It makes it a little harder to see what's going on here with the chemistry, and it doesn't look as aesthetically pleasing. The Cartoon Sidechain Helper will remove the backbone. So if I hit Set and type in CAR and hit Tab, you can see it takes the CAR and fills it to cartoon, and then type side. It fills in the rest by hitting Tab and then hit 1. It will hide the backbone to make it look quite a bit nicer. The last thing we do is remove hydrogens. Now, if you were paying attention earlier, you may have noticed that I mentioned you can actually change a setting to auto remove hydrogens when you import a new file into this. Now, if you didn't check that, that's okay. You can still remove the hydrogens by typing in remove hydro, and boom. That's actually all you need to do to initialize settings. If you look to the right of the preview window, you'll notice a box. This is called the internal GUI. In it is a series of entries, including the PDB name we selected, 1EMA. If you click this entry box, it will toggle whether or not 1EMA is shown. Similarly, if you click the All box, it will toggle all boxes. You may notice red marks around the protein. These are part of the solvent that was left over from the capture of the crystal structure. If you click one of the marks, you will see it highlighted in pink. You will also see a box on the right called Celi. This is your selection. If you click this box, it will toggle the selection on or off. If it's toggled on and you click the white space, it'll toggle it off. If the selection box is toggled off and you click something else, it will change what is selected. So if we take a look at what we have on screen right now, we have the protein that's neon green, and then we have the ligand here in the center that's also neon green. Now this certainly does stand out, but it doesn't necessarily look very professional and wouldn't look very aesthetically pleasing in a publication. So what we're going to do is we're going to color this and make it look a little nicer. The first thing to choose is a base color for your protein. So we can do that by going next to 1 EMA, which is the entry for this protein, hitting C for color, and we're going to color it light blue. Now, in further tutorials, we will go over some good color combinations and how to choose some colors for your proteins, but for now we'll just stick with light blue. And as you can see, everything's blue, including the ligand. Now, the ligand had some other colors in there, and those are now gone. Everything is just light blue. This is a good start, but we need the ligand to stand out. So what we're going to do is we're going to zoom in on the ligand using our right click, and we're going to click it to select it. And we're going to hit C for color. We're going to go under reds and deep salmon. As you can see now, the ligand is a different color and it stands out quite a bit better. The only problem here is that we only have deep salmon for every single atom type here. It's fine that the protein is one color because we really just want to represent this secondary structure as one color, but for the ligand we want to represent the different atoms. So if we go back to our selection and we hit color and we go to the by element menu, and if you see here it has HNOS and it's missing the C. All the other ones have the C in it. What this will do is it will color these atoms and it will leave the default color we chose, which was deep salmon. It will leave that as the carbons, and everything else it will color code. So if we click this, you can now see that our ligand has all the appropriate colors that it needs. Now that some aesthetic colors have been selected for the model, it's time to render it into an image. The first thing to do is to use your mouse controls to choose an appropriate pose for the model. The pose you choose will depend on what you are trying to demonstrate or the story you are trying to tell. For this instance, we are trying to highlight the directionality of the beta barrel and the placement of the ligand inside. For this reason, I've selected this side view so the beta barrel can be seen, and a view that has the ligand as least obstructed as possible. Now that the model is appropriately posed, we can render it. At the top right of the screen, you will see a button that says Draw/Ray. For quick images, we can use the Draw/Fast option. All this will do is take a screen capture of what is currently displayed on screen. For the best quality, however, we want to use the ray tracing option. In addition, we want to make sure that the transparent background is unchecked, as without this the background will not be white. There are also a lot of options here to change the resolution. We'll go more into this later, but for now we're going to leave it default. Clicking ray or slow will initiate the ray tracing. This may take some time. Now that the ray tracing is complete, you can see the Draw or Ray menu has the Save Image to File option or the Copy Image to Clipboard. I recommend you just save the image to file in case you need to reference it later. Clicking this will allow you to name the file as whatever you want. In this case, I recommend you name it something you'll remember. And that actually concludes our first tutorial on the basics of PyMOL. Future tutorials will go more into depth on ray tracing and some more advanced settings for renders. If you have any questions, post them in the comments down below, and we'll get back to them as soon as we can. Thank you.